Good afternoon. I'm Dr. Brian Shapiro, a Marketing Segment Manager at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series. It's entitled Fusing Omics and Advanced Cell Models, a New Era for Drug Discovery and Reproducibility. This webinar is presented by Dr. Ajit Singh, Chris Condilis Magnum, and Kristen O'Malley. Dr. Singh is a senior scientist at ATCC, where he is focused on providing reference-grade whole transcriptome data from the cell lines in ATCC's collection. Ms. Condilis Mangum is a senior manager, QDI Professional Services at QIAGEN, where she helps bridge the gap between bench scientists and bioinformaticians. And Ms. Kristen O'Malley is a senior field application scientist at QIAGEN Digital Insights, where she uses her experience in genomics labs in clinical, pharma, and biotech to empower scientists using software solutions such as Ingenuity Pathway Analysis, Omicsoft, and CLC Genomics Workbench. In this webinar, our speakers will present on a novel, state-of-the-art digital platform that offers comprehensive whole transcriptome sequencing data for ATCC cell lines. If you have any questions for our speakers, please use the chat function available through the webinar program. All questions will be answered as time allows at the end of the presentation. The recorded webinar presentation will be archived on the ATCC website, www.atcc.org. So with that, I'd like to welcome our first speaker, Dr. Singh.
Thank you, Brian, for the kind introduction. Welcome, everybody, for today's webinar. The title, Fusing Omics and Advanced Models, a New Era for Drug Discovery and Reproducibility. Before jumping into my today's presentation, I would like to briefly introduce ATCC. ATCC, founded in 1925 as a non-profit organization with headquarters in Manassas, Virginia, and R&D site located in Gaithersburg, Maryland, and Germantown. ATCC collaborates with and supports the scientific community with industry standard biological products and innovative solutions. ATCC is world premier biological materials resource and standards development organization. ATCC holds 5,000 plus cell lines and 80,000 microorganisms in its repository. ATCC sales and distribution in 150 countries, 20 international distributors. I have three main agenda items in today's presentation. The number one is why reference quality omics data matters. I will talk about the persistent challenges in preclinical research and exploring the importance of high quality standardized omics data sets in advancing research and reproducibility. And then I will talk about agenda item number two, introducing ATCC cell line land, building a trusted omics repository. I will provide overview of the development and goals behind ATCC curated omics data platform. And in the end, we will talk about empowering discovery with ATCC cell line land. We will demonstrate how the ATCC cell line land platform streamlines omics data exploration and supports confident data-driven decisions. Under the first agenda item, there I will talk about the common challenges in biopharma R&D. There are many common challenges in biopharma R&D, but I would like to highlight some of those common challenges on day-to-day basis. For example, finding the right cell lines for my research is a common challenge. Many cell types are not good model for the disease I'm studying, and pre-existing results are difficult to reproduce and often not reproducible. I would like to mention a publication in 2015 by Friedman et al. An analysis of past studies indicated that 50% of preclinical research was not reproducible. One of the main factors driving non-reproducible research is the use of unauthenticated biological reagents and reference materials. And relating to the biological reagents and reference material, traceability of the biological reagents and reference material is a common issue that often contributes to research irreproducibility. This problem arises due to the lack of standardized procedures for tracking the origin, quality, and handling of these materials. This schematic presentation illustrates how the dissemination of biological reagents may contribute to research irreproducibility. For example, a cell line has been acquired from the ATCC, but over time it has been transferred to various investigators and labs. And after several years, a publication been produced, but it's very difficult to track all the metadata information and the physical traceability and ensuring the physical traceability of the used cell lines. Ensuring proper traceability can significantly enhance the reproducibility of scientific experiments. So therefore, ATCC is now doing the molecular characterization of all its cell lines by performing the RNA sequencing and whole exome sequencing and making it available through ATCC cell line land. And as you can see that relating to the challenges with traceability, in addition to traceability, there are many other challenges. For example, source material challenges. There are lab adaptation. Once you receive the cells and start growing in your lab, so that is another challenge of adapting the cell culture from repository to your own lab. Secondly, untraceable physical materials because of over the period of time. Second and third, sample mix-up and missing metadata information is very common, and unknown chain of custody. Likewise, genomic data challenges also occur when you using the public data as well. For example, differences in sequencing technology in various labs and bioinformatics pipeline, very little curation from the individual labs, highly variable quality, missing metadata information, and again, traceability to the physical material from which the data is being produced. To address these common challenges occur in the preclinical research, ATCC partnered with Kaizen Digital Insights to address these most common challenges and curating a reference quality omics data repository of the ATCC gold standard cell lines. To ensure the highest standard of data quality and reproducibility, data harmonization from ATCC cell biology collection to ATCC cell line land follows strict quality assurance criteria at each step. This meticulous process results in the production of high quality reference data for each cell line, ensuring the reproducibility and traceability to the existing physical materials. The schematic illustrates the workflow of ATCC cell line land data production under ISO 9001 standards. Authenticated cell lines directly obtained from the ATCC repository are grown in the ATCC cell culture laboratory using process parameters defined for each cell line. RNA is subsequently extracted from each cell line, and its quality and quantity are assessed by nanodrop, tape station, and qubit. Each cell line contains a minimum of 5 biological replicates to ensure scientific rigor and data reproducibility. We set the 260-280 ratio from 1.8 to 2.2 and the RNA integrity number equal to or above 6.5, meeting the RNA-QC criteria required for further processing of library preparation and sequencing. Sequencing libraries are prepared using Illumina whole RNA sequencing protocols and sequenced with Illumina Next6 short-read sequencer. The produced sequencing data undergo a quality check before being sent to QIAGEN Digital Insights, making data visuals available at the ATCC cell line land. Under our efforts to produce transcriptomic and genomic reference data for human and mouse cell lines, we have completed RNA-seq and whole exome sequencing for over 600 cell lines, representing various tissue types and disease types, which are accessible through ATCC cell line land. Our goal is to complete RNA-seq and whole exome sequencing for 200 cell lines annually, with data being released on a quarterly basis. Each cell line has at least 5 biological replicates with a minimum cutoff of 3 biological replicates. What you can do with ATCC cell line land? With ATCC cell line land, you can leverage RNA-seq and whole exome sequencing data to perform various visualization of pre-analyzed datasets. Here are some key capabilities. For example, you can compare gene expression across within cell lines, explore pre-computed differential gene expression, analyze pathway and gene interactions, discover biomarkers and therapeutic targets. Based on gene expression, you can perform predict drug treatment responses, investigate molecular mechanism of disease pathogenesis, use a reference control in your experimental setting, build hypotheses and design experiments, and make data-driven selection of suitable cell lines. Now I provide some of the examples using the ATCC cell line land. For example, finding the right cell line for my research is a challenge. For example, if you have a specific gene, if you are working a specific gene and want to look at the expression profile of that particular gene across the cell line, you can easily visualize the expression of that specific genes across different cell lines. For example, there are several key genes associated with acute myeloid leukemia, and mutation in p53 gene is common in various hematological malignancy. You can look at the p53 expression in leukemia cell lines, and you can identify the cell lines shows a negative expression and positive expression. For example, the cell lines within blue box indicating negative expression for p53. At the same time, the cell lines within red box indicating the expression of p53 that is overexpressed. So you can have the selection of negative cell lines as well as positive cell lines for p53. Not only the gene expression, but you can also look at the transcript expression for your gene of interest. For example, there are several transcripts are reported for p53. And I looked at the transcript expression of p53 in Kasumi3 cell lines. And you can see that only 2 transcripts are expressed in Kasumi3 cell lines, and other transcripts are barely detected. Coding mutations are very common in various malignancies. Through ATCC cell line land, you can also explore coding mutations in your gene of interest. For example, p53 across different cell lines. In this genome browser view, coding mutation in 53 gene are highlighted within red boxes. And specific genetic mutations can make cells more susceptible to certain drugs. For example, mutation in p53 gene can make cancer cells more sensitive to p53 inhibitors. So you can select the cell lines showing the p53 mutations as well as the cell lines having the negative for p53 mutations to design your experiment and perform the drug screening experiments as well. Through ATCC cell line land, it also enables the visualization of multiple gene expression correlation across various cell lines, predict drug sensitivity response, and optimizing cell line selection. Variation in gene expression levels can affect how cells respond to drugs, and overexpression of drug targets can increase sensitivity. In this example, the x-axis indicates the number of RNA sequencing datasets produced from biological replicates of the respective cell line, each containing minimum of 3 biological replicates, and the color code purple indicates induction while dark blue indicates the reduction. Through ATCC cell line land, you can also evaluate the differential expression pattern of specific genes in normal and cancer tissue. In this figure, the mRNA expression level of BHL gene shows significant difference between normal kidney tissue and kidney cancer tissue, particularly in clear cell renal cell carcinoma. In clear cell renal cell carcinoma, the VHL gene is often inactivated due to mutation, leading to reduced mRNA expression compared to normal kidney tissue. Through ATCC cell line land, you can identify differentially expressed gene on a global scale in disease cells compared to normal cells. Chronic kidney disease affects more than 840 million people worldwide. The volcano plot generated by ATCC cell line land exhibits genes differentially expressed in polycystic kidney cell lines versus normal primary renal epithelial cells. These were some of the examples I just showed using the ATCC cell line land. And now I will hand over to Kristen to talk about the use case examples from the real world applications. Kristen.
Thank you so much for that introduction, Ajit. So I would love to take this time at the end of the webinar here to walk you through a real world example where we'll take a look at a paper that was recently published and then try to select some cell lines to study the phenomenon a little bit further. We'll then take a delve into another software called Ingenuity Pathway Analysis to have a look at the biology that is being observed in these samples. Next slide. So in this paper, what the researchers did is they took a bunch of early stage non-small cell lung cancer patients. And what they wanted to do is they wanted to try to figure out, is there a signature that will predict enhanced proliferation, which is also happens to be associated with response and better survival here. So what the researchers did is they applied some sequencing and came up with a 140 gene signature. And what they're calling that signature is this high proliferation index. So this is indicating that there is a subset of tumors that seem to be a lot more aggressive. They tend to have a lot more metastatic behavior. And they also tend to have a more immunosuppressive tumor microenvironment. Now, along with this more aggressive phenotype, it just so happens that these high proliferation index tumors also seem to respond really well to anti-PD-L1 and low-dose radiation treatment combination therapy. So with this in mind, we would love to investigate exactly what's going on with this biological finding, right? What is driving the biology that is behind this 140 gene signature? Next slide. So here is a screenshot of a heat map that was published in the paper, where we can see all 140 of the genes, which are actually plotted out along the x-axis. And then on the y-axis are all of the assorted patients that were sequenced for this particular research experiment. In the color bars over on the right-hand side, you'll see many of the metadata attached to those sample level details here. And one thing that I do want you to take away from this figure here is we notice a phenomenon of a lot of blues, those cooler colors, clustering towards the top of this heat map versus we have a lot of those reds clustering towards the bottom of the heat map. And we can see that those are pretty much connected to having a low proliferation index or a high proliferation index, right? And that is driven off the fact that those low PI index samples all tend to have more downregulation of those genes in that 140 gene signature versus those high PI index patients seem to have more gene expression or a higher amount of gene expression for those 140 genes here. Next slide. So now the question lends itself to be asked where, if we wanted to study what is going on with those 140 genes, how are they driving this more aggressive phenotype in these lung cancer samples? And why are they responding so well to both radiation therapy as well as IO treatment? Well, what we can do is we can have a look into the QIAGEN OmicSoft ATCC cell line lamb that Ajit described earlier in this presentation, and we can narrow in onto lung-derived cell lines here. So all along the y-axis are a whole smattering of different cell lines, like I said, derived from lung. And then on the x-axis are all of the genes that are contributing to that 140 gene signature for PI tumors here. What we can see with the next slide is in this teal box that's showing up on the heat map, we can zero in onto an interesting cluster of cell lines here. So you'll notice that we have a good stripe of blue at the top indicating downregulation of these genes, and a more kind of red stripe towards the bottom of this teal box, indicating that we have more upregulation of these PI tumor signature genes. Next slide. So zooming into that teal box, what we can see is once again, we have our cell lines on the y-axis and all of our genes plotted out along the x-axis. And we can see a really striking difference between some of those cell lines that seem to be kind of recapitulating what we see in those low PI gene signature tumors versus a smattering of cell lines that have a high proliferation index to them. So at the top here, all of those blue cell lines could be used to study ways to perhaps introduce a more immune enriched environment here. Versus towards the bottom, those are going to be cell lines that are really reflecting those same phenotypic outcomes of those tumors that we saw in the paper there, right? So that's enhanced proliferation, really more aggressive type of a phenotype. So what we can do is we can take those cell lines, next slide. We can take all of that data that we're getting from OmicSoft for those cell lines, and we can deposit it into another software called Ingenuity Pathway Analysis Software. What this software does is it's a network analysis software that leverages something called the QIAGEN biomedical knowledge base. And what that is it's a collection of over 20 years of knowledge that we have gained through combing through things like literature, repositories, databases, to find out things like how are genes connected, how do they influence different outcomes or functions, things like that. So we can utilize IPA and that knowledge base to build a network of these 140 gene signature genes from that high proliferation index. So here what we're looking at is actually a connected network of a good amount of those genes from the gene signature and how they play together biologically. So we can have a look to see how are these genes connected, what are they influencing? And then take another step and say, all right, what happens to these genes when we overlay data from OmicSoft to see, if I have a high proliferation index tumor where everything seems to be increased in expression, what does this particular network look like? We can then again ask systems biology questions like, what canonical pathways seem to be enriched, right? What are the things driving some of the biological outcomes seen in these patients? So first things first, we can see by that annotation that says cell cycle checkpoints with that pink CP acronym next to it. What this is showing us is that we are heavily enriched for a canonical pathway called cell cycle checkpoints. It's pointing to all the constituents of this particular network that are showing up in our data here. Now, this is actually rather interesting to know because, as we all probably know, immuno-oncology type therapies seem to go after these cell cycle checkpoints a lot. So it's no wonder that we see this as enriched in that gene signature. We can also do many other things, but one of the other facets that I wanted to show off here is the fact that we can look for other diseases, other functions enriched in this particular network. So here by that green FX lung adenocarcinoma annotation added into the network here, we can see that indeed this network is heavily enriched for lung adenocarcinoma driving genes. So it's a nice sanity check to show us that we are indeed on the right track with this gene signature and what it does to influence the biology of our non-small cell lung cancer patients. So all in all, at the end of the day, you can see how we have a really nice back and forth between Omicsoft, where we can farm for the data, have a look at different cell lines that possess interesting gene signatures or different mutations that we might be interested in, and then plug it into Ingenuity Pathway Analysis software to check out the biology and get more information about behind the knowledge of the biology of what's going on, in these systems to drive these more aggressive cancers. Next slide. I'd love to hand the reins back over to Ajit to close us out for today.
Thank you, Kristen, for the nice presentation. And I would like to provide the summary of today's presentation. ATCC Cell Line Land is a trusted omics data resource built on authenticated ATCC cell lines, offering traceable and high-quality reference data sets. By integrating genomic data with authenticated biological materials, the platform drives innovation and supports faster, more reliable drug discovery and development. All data is generated using a standardized ISO 9001 compliant workflow to ensure scientific rigor and reproducibility. The platform is continuously expanding with over 200 new cell line data sets added each year through quarterly updates. Thank you for your attention. And the take-home message is ATCC Cell Line Land, your go-to resource for answering the most critical questions in preclinical research with confidence, clarity, and credibility. And we would like to answer any questions you have.
All righty. Well, thank you for the excellent presentation, Ajit, Chris, and Kristen. In just a few moments, we will begin our Q&A session. Please remember to use the chat function available through the webinar program to submit your questions. This session will be archived on the ATCC website at www.atcc.org. And it looks like we've had a few questions come in already, so let's go ahead and jump in. For our first question, does Cell Line Land pick up non-ATCC cells? I guess I'll go ahead and throw that one Ajit's way.
Thank you, Brian. ATCC cell line land is mostly focused for the cell lines deposited in the ATCC repository, and we are primarily focusing on human cell lines and mouse cell lines.
Alrighty. Do you cover anything besides just Homo sapiens and mus musculus?
At the moment, we are focused on human and mouse, but if someone is requesting some other species, we also can consider to sequence those cell lines as well.
Alrighty, great, great. Now, how often is cell line land updated? And I can toss this one up to either Ajit or Chris.
Chris, can you answer that? How often cell line land can be updated?
Yes, ATCC cell line land is updated quarterly with about 250 individual samples, which correlates to about 80 new cell lines per release.
Now, I'm curious about those individual samples, and I'm just sort of extending this question a little bit. Is that for individual production lots or batches you can get that information for, or is it just like from one token lot?
It is about.
Oh.
Go ahead.
So part of our mission to create a resource that has great traceability, we actually have the metadata for each of our samples, which lot they were taken from and when. Each sample is sequenced independently, and for each cell line, we have around five individual samples, typically from the same lot. Over time, we plan on resequencing the most common cell lines that are in production to take into account any genetic drift or changes that can occur, through culturing standard practices.
Very nice, very nice. All right. So this next question kind of addresses another major cell type that's out there, primary cell. So, both Ajit or Chris, you could handle this one. Do you have primary cells in cell line land?
Yes, the cell line lands includes primary cells, continuous cells, engineered cells, all different types of cells from the repository. Has been generated from any tissue types or disease types.
Great. So some of the newer cells that ATCC has been developing lately, like with the luciferase reporters or some of the isogenic cell lines that were generated via CRISPR gene editing, then those are also included?
Yes, so those cell lines are also being included as well.
Okay, great, great. And then about how many tissue types are represented in cell line land?
So we have completed several tissue types. For example, we have completed blood, we have completed kidney, we have completed breast, we have completed lung, and as of now, we are sequencing brain tissue cell lines.
Okay, great, great. So then, what's overall the roadmap look like? Are we trying to get everything into cell line land? Every tissue type that's available at ATCC?
Yes, so the goal is to sequence every tissue types and the human and mouse cell lines. So currently ATCC has over 2,000 human cell lines and as well as like several hundred mouse cell lines. So our goal and plan is to sequence every human and mouse cell lines available in the ATCC repository.
All right. And, I see that a few people have joined kind of late and are kind of asking this question. what types of data are available? Is it just expression or beyond that?
Yeah. Chris, can you answer?
Yes, so we started with expression data, but we are also adding in whole exome-seq data. The goal is to have a paired whole exome-seq data with every new cell line that we added. We're currently going back and kind of backfilling for the initial releases of data that were added to the resource.
All right, good, good. Oh, we've actually had quite a few questions coming in still. So this next one, can the links for the data sets and softwares be provided via email or a workflow after the presentation? And actually, that's one that I can kind of take. Yeah, we'll be sending out communications to the folks who registered for the webinar after, and they will certainly contain the links to you know, the data. All righty. And then let's see. Okay. And this next question, have you analyzed any data similar to the cell lines you sequenced and publicly data regarding the same cell lines? What type of changes did you see?
Yeah, I can take that one.
Oh, sure.
Yeah.
Yes, so we used our standard pipeline that we use for some of our other land products, which includes disease land and oncoland. And in those repositories, we've done similar sequencing experiments from public data. Granted, the data has been taken from the raw form, just like we have here, processed with the same profiles. And actually, Ajit has done some comparisons that he's presented at various meetings. We do see that there are some cell lines, you know, from the same background that do correlate with each other. Their expression pattern is very similar. If you do a PCA plot, they do cluster with one another. But I think Ajit and I have both identified that there are certain public data sets where the profile is quite different, regardless of treatment. You know, we kind of pre-filtered for untreated cells to do this comparison, because that's what we've done here for the cell line collection for ATCC. So there are some differences, and there are some genetic differences as well. And so we do see that there are mutations in those other lines that we do not find in the lines that we've been sequencing directly from the ATCC repository.
Okay, great, great. This other question I can kind of answer around the first research paper that we showed during the presentation. And that was the, let's see, the Cell Reports Medicine paper, signature of enhanced proliferation associated with response and survival to anti-PD-L1 therapy in early stage non-small cell lung cancer. That was volume 5, issue 3 from March 19, 2024. Open access journal. The first author is Nasser Al-Torki, and the corresponding officer is Tim McGraw of Corning Wheel. All righty. And then let's see, we have one final question that's come in. So let me sort of do last call for questions before I jump into this. So please go ahead and submit your questions if you have any more. Could I know what's the difference between lot-to-lot numbers of cells of the same cell lines, numerically speaking? Ajit, do you know the answer to this question?
Yeah, that Very interesting question. So we haven't compared lot variations that might be existing for the same cell lines, but that is also under our radar to sequence different lots and look at if there is any difference between the lots for the same cell line.
Okay. All right. So we have not seen any additional questions come in. I guess with that, we can close the Q&A session. So I'd like to thank Ajit, Chris, Kristen for the excellent presentation, and thank you everyone for attending the webinar. The video and PowerPoint presentation will be posted on the ATCC website, www.atcc.org. Thanks again, everyone, and have a great day.